Okay, hello guys. So first of all, I would like to thank the organizer to invite me to become speaker in this meeting. So my name is, how we should do this? Okay, my name is Juliandi from Bogor Agricultural University, Indonesia. This is our campus. It's very nice and situated near the Mount Salak. You should visit it anytime if you want. So Jenny already pointed out that during our life, from the embryonic stage until adult, we sometimes need some cell to replace the missing cell. So I want to show you the proof of that, which is if it is not going very well. So this is me before taking my PhD course. So as you can see, many cell loss happened after I taking my PhD. So yeah, I'm kidding. This is not me. So, so this is one of my heroes, Superman. So the actor is very famous called Christopher Reeve. Maybe you already know. This is like my hero during my childhood. So I really like to watch this movie. But unfortunately, the late Christopher Reeve, before he died, actually he suffered some kind of accident while he taking some sport. And then he fell from the horse. And then he suffered what we call spinal cord injury. This is very bad. And the cause of spinal cord injury in the US, this is the data from National Spinal Cord Injury Statistical Center in the US. So mostly by some accident using the car or truck or something, and then falling from somewhere, not falling in love, obviously, and then some violence. Okay, so we really concerned how we can cure this injury, because this is really the main injury also in other country. So the problem in the spinal cord injury, if you have an injury in the spinal cord, it will lead many inflammatory cytokines. Then it make the neurostem cell which reside in the spinal cord injury to become astrocyte. So more astrocyte produced than neuron or oligodendrocyte. But actually, to cure spinal cord injury, we need more neuron and more oligodendrocyte. So then we kind of thinking something to solve this problem. As Jenny already mentioned in her talk, that neurostem cell is self-renewing and can give rise to neuron or astrocyte or oligodendrocyte. So what control this? One is internal program within the cell, and the other one is external cues. These two things, it's controlling what they will become, what is their fate. So one of the internal program is the epigenetics, okay? The program within the cell, epigenetic. So I only will show you one example of this epigenetic mechanism, which is histone modification called acetylation. So if you still remember the biological course in your high school or in your college, so DNA is negative charge, okay? And this histone have a tail which is have a positive charge. So they really like to bond each other, okay, in normal condition. So the protein which is regulating the transcription cannot bond to their binding site. So like this one, it cannot bound to the motif which is they usually bind. But if you have acetyl group attaching to this positive histone tail, then the histone tail will lose the positive charge, making the chromatin structure become relaxed. So then it opens the binding site of this protein, so then this protein can attach to the DNA and then regulate transcription. So this is kind of off, and this is kind of on state. So this process is catalyzed by two different enzymes. First one is called histone acetyl transferase, which is catalyzing the attach of the acetyl group to the histone tail. And the other one is histone deacetylase. We call it HDAC. Okay, so if I say HDAC, it means the histone deacetylase. And it removes the acetyl group from the histone tail. This process is like very dynamic and vice versa. So we can control this by increasing the acetylation or increasing the acetylation. In my concern, I want to increase the acetylation. So we can do it by two ways, by increasing the HAT activity this way or inhibiting this pathway. So I choose the easiest way. We just inhibit the HDAC or the acetylase. So how we can do this? So in the medical practice, it's already been done actually. So there is a drug called Valproic acid. This is an established drug in long treatment of epilepsy. So it also have a HDAC inhibitor property. So we can use this drug or this chemical to inhibit HDAC. And Jenny, with her friends, already show very fabulous paper, which is the most cited paper maybe for this field in the histone acetylation. Because in theory, like I said, if you inhibit HDAC, then you open the gene. Then you will expect that neural stem cell will have the neuronal gene open, astrocytic gene open, and the oligodendrocytic gene open. Then you will have a mixing of neuron, astrocyte, and oligodendrocyte if you inhibit HDAC. But apparently, when Jenny and her friend tried this in vitro using neural stem cell from the adult hippocampus that she mentioned earlier, it turned out to be neuron only. They cannot find astrocyte or oligodendrocyte. And then, even if they induce by astrocytic cytokine, in control you can see many astrocyte is produced. But if you put VPA, you inhibit the HDAC, then you can only find neuron. And then it's the same when you induce with oligodendrocytic cytokine, and they become oligodendrocyte, but if you put VPA, they become neuron. So this is very specific neuronal differentiation by inhibiting HDAC. So in our lab, we are very interested with this paper. Then we ask the question whether the different source of neural stem cell will give different result. So we then use the embryonic forebrain neural stem cell. So not adult hippocampus neural stem cell, but more earlier, embryonic forebrain neural stem cell. So we just cut the embryo of the mice and take out the neural stem cell. I'm sorry for you who love animal, I just have to do it. So then we just cut, and then we culture this neural stem cell. Oh, I touched something. Okay, sorry for that. And then after we put VPA, as we found earlier, it become more neuron. Okay, but slightly different result. If we put this neural stem cell from embryo to astrocytic condition, it's not just make neuron like the adult hippocampus, but it repress the astrocytic differentiation. And it's also true for the oligodendrocytic condition. If you can see this oligodendrocyte, but using VPA, it become repressed. So I want to remind you about that spinal cord injury environment where they produce more astrocyte, but actually we need more neuron. So we kind of hoping that this VPA-induced neuronal differentiation will benefit for the spinal cord injury. So then we use mouse, which is we already put some marker, which is here is GFP into their neural stem cell, and culture this neural stem cell, so we can distinguish the neural stem cell that we transplanted with the host cell, which is endogenous in this mouse. So we just open the spinal cord and we make some contusion or to break the spinal cord of this mice. So what happened if we do this? So you can see this is 1 week after we make the contusion or spinal cord injury. They completely paralyzed, the hind limb completely paralyzed. They cannot walk by the hind limb. If we do nothing, after 6 weeks, they still paralyzed. if we transplant with neural stem cell only, they can move a little bit, but they have to drag their hind limb, so they cannot support their weight. Okay. Then we use neural stem cell and the VPA to induce more neuron. So as you can see, they can walk as like the normal mouse, if it is not even, how to say, almost perfectly normal. So they can support their weight and walk again. So this is the mouse cell transplanted into mouse. So we are very excited with the Takahashi and Yamanaka founding that Jenny already mentioned, that we can actually use another cell type, because it is impossible. If you have spinal cord injury, then you have to take your neural stem cell from your brain or your spinal cord. It's like you curing something, but you destroy another thing. So then it will be helpful if we can use another cell which is already differentiated. So like Jenny said, we can use the skin. So then we can take this skin and reprogram into the what we call iPS cell, which is have ES cell or pluripotent state, and we can direct them into many cell type. In this case is neuronal cell type, and then we transplant back to the patient. So that if someone has spinal cord injury, we just take the skin. So we don't need to destroy the other nervous system. And we did it. So but we don't use human. We just take the skin of the human, and then we redirect them into the neural stem cell, and we transplant it into the mouse. So this is mouse after spinal cord injury. So completely paralyzed in hind limb, and this is 8 weeks after. They can move, but they have to drag their feet. And using the human skin derived neural stem cell from iPS, they can walk again after the spinal cord injury. So we are very happy to found this, and we trying to make another progressive research after this. So that's one part of my talk. So another part is I want to remind you again about the stem cell. So Jenny already explained about the zygote and the big ball with the inner cell mass, which we call it embryonic stem cell. So please remember this embryonic stem cell, and how this embryonic stem cell can become another type of cell such as neural stem cell. So the question also interesting is, how about neural stem cell which is come from the ES cell? So not from the embryonic forebrain, but from the very early ball in the embryo, before the embryo. So actually we already know so much about how to direct this ES cell to several types of neuron. So for example, we can derive this ES cell to become neuronal lineage, and we can make many types of neuron by using cytokine or drugs. We can make cortical pyramidal neuron, ventral forebrain neuron, spinal cord neuron, and so on. So many types. It's already known. I'm interested only to use this pathway because this pathway leading to the differentiation of cortical pyramidal neuron. And why is that is so interesting? Because it composes the cerebral cortex. The cerebral cortex is very unique because it's help you to have the good cognitive function, and also help you to have the consciousness. And why it is also important, because, for example, in the mammals, you can see mouse brain is very small, and the size of cerebral cortex is like size of stump. And in chimpanzee, you can see there is many folding in the cerebral cortex, which is increasing the size of the area of the cerebral cortex. It is like a A4 size area. And in human, it's even more complex, more folding, and it is like the 4 times of the A4 size paper. So why human during evolution have the very increasing brain size of the cerebral cortex? One of the reasons is because in the mammals, they develop the layering of the neuron in the cortex. So as you can see in amphibian and the reptile, they don't have layering. They only have neuron which is directly connect to the motoric function for breeding, for the normal animal trait to survive. But in the mammal, they have layering, which is, for example, the layer 5 and the 6, it's project to the spinal cord and another area which is important for basic function of the behavior. But this layer 2, 3, and 4 is project contralaterally to the other hemisphere or locally to make you conscious and can learn more and more have cognitive function. So we think this is very important layer. We call it this layer 2, 3, 4 as the upper layer, and we call this layer 4 and 5, sorry, layer 5 and 6 as a deep layer because of their different layering and different function. And as you can see, more advanced during the evolution, the layer is become more complex. So how we distinguish this neuron? So each of these neurons express protein which is different. So as you can see here, they express different protein. For example, layer 2, 3, and 4 express the COX-1 protein, and layer 5 and 6, they express the CTIP2 protein, and the layer 1 express the reelin protein. So we can just use some antibody to recognize this protein and give some fluorescence of this antibody. So then we can know what layering. So then, like I said, we interested whether VPA can also enhance neurogenesis of the ES cell-derived neural stem cell. And as you can see here, it also induces more neuron by looking at these two neuronal marker. It's induced almost 2 times of neuronal differentiation. So confirming that histone acetylation is important in neuronal differentiation. And like I said, we want to know the type of neuron produced by using the VPA. We can see that VPA represses the deep layer neuron. So this is the green dot is the CTIP2 positive deep layer neuron, and this red dot is the reelin layer 1 positive neuron, and some of them is co-positive for both marker. And VPA represents almost half of them. In contrary, if we use VPA, it's actually increasing the COX-1 positive upper layer neuron. And if we quantify it, it's almost 4 times increase. So this is very exciting because we then can model that how the mammal acquire the upper layer during the evolution by using this model. And so I will give you the summary of this second talk. That we know that neural stem cell produce deep layer CTIP2 positive neuron, and later on they will make the COX-1 positive upper layer neuron, and this is enhanced by VPA. And histone acetylation is important for production of neuron in vitro. So next question is, how about in vivo? So we just use this drug during the pregnancy of the mice, and it is already known that if we put early in pregnancy, it's going to be not good. And if we put in the mid or late gestation, it's no problem, they think. So then we just use these mice, pregnant mice, and give VPA during day 12, day 13, and day 14 of the pregnancy of the mice. And we cut off the brain and see whether it increased neurogenesis, and we found it is indeed increasing neurogenesis. And in brief, it also increasing the upper layer of this neuron in vivo, and also decreasing deep layer neuron in vivo. And the second summary is, we know that now that also the neural stem cell produce deep layer neuron and later produce the upper layer neuron, and VPA somehow increasing this progressiveness and implying that histone acetylation is important in vivo. So then, is there any consequence of this event after they become adult? So in brief, we we put VPA during pregnancy like the previously, and we tested the behavior after they become adult. I cannot discuss one by one this behavior test, but in brief, we found that learning and memory have some deficiency. So I'm gonna show you one example of the learning and memory that we done. We just put the mice into the cage and we give a tone. And after the tone, we give electric food shock. So if the mice clever, they can know that this box is dangerous and this tone is dangerous because it gives food shock. And second day, we just put them in the same box at day 1. We just measure how many freezing or scary they are in this box. And the day 3, we put into different box, but same tone, and we measure how many times they are freezing or scary. And basically, VPA mouse perform less. And already shown by Jenny that the learning and memory is controlled by hippocampus. So this one, the hippocampus part in the mouse, we can label the neural stem cell by injecting some chemical which is absorbed by replicating cell. So we put the BRDU into this mouse, and we can label the neural stem cell in the hippocampus. So you can see maybe here the hippocampus have less number of neural stem cell in the VPA mouse. And the next question is how we can overcome this deficiency, because we have many children born from the epileptic patient. So we just give them to run. And the VPA mouse, after running, and then we measure again number of neural stem cell by BRDU. As you can see, the number of neural stem cell is increasing. But is it really recover the behavior? We conducted the same test, and we found it's not recovering. But in the skew conditioning for recognizing the tone, it recovering after running. So then we want to make sure. So we did another test, which is more dependent on hippocampus. So mouse have to mouse have to walk from A to B to C. So each walking have to be new arm. If they go to the different, the arm that they already entered before, for example, A, B, and then go back to A, it is considered to be wrong. So we just measure how many correct alternation that they make. And apparently running helped the VPA mouse to perform better. And this is the last summary. So VPA treatment during embryonic stage in the mouse, and make the neural stem cell, the adult neural stem cell become sedentary, and then make lower proliferating, and then make lower number of neural stem cell, and it leads to learning and memory deficiency. But if we put them into running, it recover the neurogenesis and also leading to the recovery of learning and memory. So this is the take-home message or point. First, I want to make sure you know that now we can cure spinal cord injury in mouse model by transplantation of epigenetically modified neural stem cell or reprogram human skin cell. And then we can induce more upper layer neuron, which is important for learning and memory also, by increasing histone acetylation, both in vitro and in vivo. And then we can cure deficiency in learning and memory of children from epileptic mother, at least in mouse, by just let the children running voluntarily, not obligatory. So I want to acknowledge some people from Bogor Agricultural University and NICE Japan and many other universities which is helping me doing this research, and thank you for your attention.